Hi, my name is Nyabutiam. I'll be representing the Tangu Kale Labs team, and the rest of my team members won't be able to join the presentation, but I hope I do them proud. For a long time, I've been working on building tools for microbiologists in Kenya as someone who's an actual microbiologist, but over the years, I've learned how to code and particularly look into data analysis, and right now I am a data scientist. Enough about me. Uh, my team is working on making the lab a better place, particularly looking at the processes that happened in the lab, such as microscopy, which is considered a gold standard for some diagnostic tests. And our mission is to improve efficiency and accuracy of labs worldwide. So our target audience is laboratory professionals, and particularly technicians and managers in hospitals. We've realized this particular group of people is highly skilled, but for the most part they are overworked, and they usually value efficiency and accuracy in the processes that they normally go through day by day, and they are also tech savvy. So what happened is, looking at all these factors, we created tools particularly for them, and they mostly span within microbiology and parasitology terms, just study of microorganisms. And we created three things. One, we created a computer vision powered machine learning system. So what that means is you just give this, the computer is able to figure out what's in an image and be able to tell you with a certain amount of accuracy that this image has this particular cell. And another thing was protocols. So protocols are just processes or workflows these laboratory technicians usually do to be able to ascertain that, for instance, this person is suffering from malaria. Uh, so that's a protocol management system. Then for the epidemiology bot, that one is actually, first of all, epidemiology is just the study of health related states within a particular place. So maybe a couple of people are having malaria. It's high time now to look into that and kind of figure out what is causing this malaria, as an example. So the early adopters of these particular tools we have been working on say that this tool allows easy identification and quantification of parasites. That is the first tool that I've just pointed out. But without further ado, let's just look at the tools. So this first tool, I have uploaded an image of cells. These are with a couple of these cells are red blood cells, so I've just uploaded here. So let me just clear it so that you can see. So let's just upload it again, and then if I click on submit, the computer will be able to identify what's in the image and say, oh, there's a red blood cell in there and a couple of the causative agents of malaria, which is plasmodium. So these are the RBCs, and these are the red blood cells, and this is the cells that are infected with plasmodium, and this is the accuracy. So it's very accurate, that it's very sure, or this model is very sure that, oh, there's a high likelihood that within this image that there are red blood cells, and particularly having like these infections of this is actually a stage in the life cycle of this plasmodium called the trophocyte ring stage, and a general trophocytes. So majority of the cells here are infected, and this is super important for this lab professionals. As for the laboratory guide or the lab protocol guide, what I did is I've uploaded a couple of my documents. They have been changed into a representation that's easy for search and retrieval. And what I've asked is, how do I perform gram staining procedure? And for each step, add an emoji. And then the particular model was able to tell me, oh, this is how you do it from your documents. So it just referenced my documents, which have many sub documents, and we are able to we're able to get the gram staining procedure, and this is actually correct. And I also appreciate the color purple being shown here and the color red. This is very important in differentiating cells after doing this particular procedure. So yeah, this tool is pretty nifty. And lastly, we have this one for where you just look at the date of interest, which is 2024, and then you'll be able to, this particular tool will be able to go and gather information about what's going on when it comes to disease outbreaks, and then it'll be able to come up with a report like this and present it like a epidemiologist would, where they cover the where, when, why, what, and who. So basically, this one is talking about a measles outbreak in Nigeria, and we also have a measles outbreak in. We don't really have a measles outbreak, but we have a vaccination shortage in Machakos. That means that particular area, the children are vulnerable to getting the measles, and measles is actually something that can be avoided just by vaccination. But tools like this help us with just being prepared for outbreaks, and COVID has definitely taught us that systems like this would be useful for, like, as alert systems to tell us to prepare better. So those are the tools I wanted to show you, and I hope whoever who's watching this video has found these tools useful or helpful in some way. And if you want to be part of our story in any way, like maybe you want to be part of the team, or you want to help us with maybe compute or hosting services, you are very much welcome. And if you want more information, you can either scan the QR code over there or use the forms link over there to contact us. So what's next for us? So obviously I'm gonna make a comprehensive landing page for you to get these tools that we've mostly put in Hugging Face, and also improve the cell identification tool to now be able to count the particular cells, and then lastly make the epidemiology agent faster and be able to, like, alert people a bit faster, like, based on information gathered online, like, this is what's the most likely outbreak that can occur in this particular country, and here we're looking at Kenya. So thanks for watching. See you soon.